All right, our final Unit 3 topic for review is the cell cycle. All cells have to undergo three basic processes associated with reproduction and division. First, they have to regulate their DNA. That usually happens in what's called the S or synthesis phase of interphase. And it has to ensure that each cell gets a complete copy of the DNA. Next, the replication of any items that are in the cytoplasm, like the ribosomes and the membrane-bound organelles. And then finally, the division of the cytoplasm and cell membrane. We commonly call this cytokinesis. So bacteria perform this process in a process called binary fission, or sometimes prokaryotic fission. It's a type of asexual reproduction. This process doesn't create any kind of genetic diversity because it's essentially a type of cell cloning. So the DNA, the replication process starts at one origin of replication on the single circular chromosome that the bacteria have. Process works its way around that entire chromosome, results in two identical chromosomes. A cleavage furrow then forms in the cell membrane. The cell membrane splits in two, each getting an identical nucleus. The two cells that result are 100% identical clones of each other. And biologists think that binary fission eventually evolved into the process of mitosis as we know it today in eukaryotic cells. So the cell cycle is the life cycle of a cell. It includes the time when a cell is doing its job, when it's growing, and when it's replicating. The phases of the eukaryotic cell cycle, first of all, include interphase, which is the time between cell divisions. Most cells spend about 90% of their time in interphase. This is when they're working, when they're doing their job, they're doing their everyday activities, they grow, they repair, and so on. Typically, we divide interphase into three main subsections. The first is G1. During this stage, again, everyday growth, everyday activity and work, everyday repair. There's a checkpoint during G1 toward the end. A checkpoint is one of several points in the cell cycle at which the progression of a cell to the next stage can be halted unless the conditions are favorable. The cell only passes through this G1 checkpoint if it's the right size and if it has enough energy reserves. At this point, the cell also checks for DNA damage. A cell that has DNA damage, doesn't have energy reserves, or isn't the right size, it won't progress any further. Typically, it might go through apoptosis, or it might wait until those conditions develop. Either way. But if a cell checks all those boxes, it will proceed to the next phase of the cell cycle, which is the S phase, the synthesis phase. This is when the DNA is replicated or synthesized. In humans, all 46 chromosomes are replicated. Then we go to G2. This is a secondary growth phase. During this stage, the organelles enlarge and they get replicated, and the newly synthesized DNA is checked for errors. There's a second checkpoint at the end of G2. This checkpoint bars entry into mitosis unless certain conditions are met. So again, the cell assesses, is it the right size? Does it have the right proteins available? It also ensures that all the chromosomes have been replicated. There's no mistakes or damage. And if all those things are met, check, the cell will enter into the mitosis stage of the cell cycle. So all of these stages represent interphase. G1, S, G2. And remember, we talked about a checkpoint at the end of G1 and a checkpoint at the end of G2. Next, the cell will go into mitosis. Mitosis is the division of the nucleus, so it's the division of the DNA, and it's followed by cytokinesis, which is the actual division of the cytoplasm and the cell itself. So as I said before, mitosis is really not cell division, it's nuclear division. And it's followed by cytokinesis, which divides the cell. We'll talk about four main stages of mitosis. We use an acronym, PMAT, to remember those. Prophase, metaphase, anaphase, telophase. So during prophase, the nuclear envelope is disassembled, broken down. Organelles called the centrosomes start to construct the spindle fibers. The DNA, which is typically in the form of chromatin, uncondensed, unwound DNA, starts to wind up and form chromosomes. And the chromosomes that we see are typically X-shaped chromosomes. We call those bivalent because in reality, each half of this is an identical copy of the other half. We call them chromatids when they're connected. Eventually, though, those two are going to disconnect, and at that point, they'll actually be called chromosomes. The centrioles move toward the poles. The spindle fibers begin to attach to the chromosomes at a point in the middle. This middle part is called the centromere. A piece of that is called the kinetochore. Okay? So the spindle fibers are essentially going to attach there. They're going to be like little ropes almost that are going to pull those chromosomes where they need to be. There are also proteins called motor proteins that help to pull the chromosomes along the spindle fibers. And they use ATP as their energy source to do this pulling. Next, we get metaphase. Meta means middle. So at this point, the chromosomes are pulled to the middle of the cell and they're lined up single file. The third checkpoint occurs at the end of metaphase. So this is checkpoint number 3. Again, the cell checks for all those normal things, cell size and so on. But here it also checks to make sure that all the chromosomes are attached to the spindle fibers and that they're all lined up in the middle. The cell cycle can't keep going, it can't proceed until the kinetochores of each pair of chromosomes, or each pair of chromatids, I should say, are anchored to a spindle fiber. Finally, we get anaphase. So during anaphase, the spindle fibers shortened and they pull apart the X-shaped bivalent chromosomes. Some get pulled this way, part get pulled this way, and they separate. Once they separate, the chromatids are now called chromosomes. This set of chromatids is identical to this set, and they're getting pulled toward opposite sides of the cell. And again, motor proteins help to pull those chromosomes toward opposite sides of the cell. Finally, we get telophase. Telophase means means last. So those chromosomes are pulled to the opposite sides. And essentially the events that happen in prophase get undone during telophase. So a new nuclear envelope is built around each set of DNA. The chromosomes unwind and go back to the chromatin stage. The spindle fibers are broken down and the cell's preparing for division. So telophase ends with 2 identical nuclei still inside one cell. Telophase is followed by cytokinesis. It's this point where the actual cytoplasm and cell divide, and we end up with one cell, or 2 cells rather, from the initial one, and those 2 cells are identical to each other. Mitosis, like binary fission, is a type of cell cloning. One cell is cloned into 2 identical cells. 2 identical cells that are just like each other, and just like the original cell they came from. So another stage we'll mention is the G0 phase. And this is called the zero growth phase. It's a period in the cell cycle in which cells exist in what's called a quiescent. It's kind of an inactive or dormant state. It's kind of like an extended G1 phase. The cell's not dividing or preparing to divide, it's just there. Sometimes it's referred to as a post-mitotic state. Since a lot of the cells in G0 are non-dividing, they're kind of outside of the cell cycle. Nerve cells and heart cells usually become post-mitotic when they reach maturity. They continue to work, but they stop dividing. Some multinucleated muscle cells don't really undergo cytokinesis either, so they're sometimes considered to be in this stage. So let's talk a bit about the regulation of the cell cycle. So one of the ways the cell cycle is regulated is through all those checkpoints we talked about a minute ago. Those are important. Regulating the cell cycle is crucial for normal growth and normal development, And the regulation can vary from cell type to cell type. But one of the most important ways of regulating it is through protein-based cell signals called cyclins. The cyclin concentration in a cell increases from the S phase of interphase all the way up until anaphase. As the cyclin concentration is increasing, cyclins combine with an inactive enzyme called cyclin-dependent kinase. You'll hear a lot of people call that CDK. Once the cyclin and the CDK get together, they form an active enzyme called MPF, maturation promoting factor. MPF actually causes cells to enter mitosis by phosphorylating the nuclear lamina, the structure that actually holds the nuclear membrane together. After mitosis is over, the cyclins are degraded. Only the CDK is left behind. It's inactive. So essentially, it's like resetting the clock. Here we can sort of see the events. So here's the cell. Here are the different stages of the cell cycle up top. We're looking at MPF activity in black and cyclin concentrations in blue. So we can see to begin with, mitosis just happened right in here. So the cell enters G1. And from there on out, the concentration of cyclins in that cell increase throughout S to G2 till they get to some critical level. At that point, the cyclins activate CDK. MPF helps to start the breakdown of the nuclear envelope, and mitosis starts. As mitosis completes, the cyclins are broken down, and the cyclin concentrations drop back down near zero. So now we've got two new cells. G1, S, G2, those cyclin concentrations go up. They get to some critical level. They activate MPF. MPF causes the breakdown of the nuclear envelope, starts mitosis. Mitosis ends, cyclins are degraded. MPF starts functioning. Cyclin concentrations start to rise in the two new cells that are formed. So one of the main ways to regulate the cell cycle is through cyclin concentration. Cyclin concentration is very low at the beginning of a cell's life. It increases throughout G1, S, and G2. And ultimately, when it releases a critical value, it causes MPF to become active, which stimulates mitosis to start. And at that point, once mitosis is over, the clock is reset. Cyclin levels drop back down to zero or near zero, and the life cycle begins again. Last thing we'll mention here is cancer in the cell cycle. So cancer is essentially when the cell cycle is not being regulated. Oftentimes in cancer, cells have abnormally high cyclin levels, which means they're dividing all the time. Also, the checkpoints typically don't exist within cancer cells. Cancer cells are often considered to be immortal because they continue to divide forever. Most cells stop dividing at a certain point. Normal cells have telomeres at the ends of their chromosomes. And every time a chromosome replicates, those telomeres shorten. And when the telomeres get to a certain shortness, that essentially signals apoptosis. And the cell dies and therefore can't reproduce anymore. Cancer cells oftentimes have an enzyme called telomerase, which continually elongates the telomeres, which means they never shorten, which means they never commit apoptosis, which means those cancer cells can continue to divide essentially through eternity. All right, that's it for Unit 3.